everyone, my name is Madeline Smith and I am a senior dietetics student here at the University of Southern Indiana, and today I'm going to be giving a presentation over the Metabolic Pathways project, which essentially is taking 11 metabolic pathways that we learned about in class and explaining to you how they're all connected. I do plan to turn my phone screen around just because I think you'll be able to see my map more up close. So yeah. Okay, so starting out, I'm going to explain the key that I have here. So the yellow highlight indicates niacin, the dark blue riboflavin, the pink highlight CoA, and the orange highlight thiamine. And then over here I have different colored arrows for the different pathways, and the reason why only some of them have colors is because I only used colors on the ones where the product left the pathway. So some of them are connected, obviously, but they just didn't start there, so that's why they don't have their own individual color. Okay, so we're going to start with glycolysis. We're going to start with pyruvate down here because that's a few. The pyruvate comes up here, and the enzyme pyruvate dehydrogenase complex, which is highlighted in orange to indicate thiamine because it requires TPP as a cofactor, and then it essentially turns into acetyl-CoA, which starts up fatty acid synthesis. And then also with pyruvate, we have it connected, follow the blue line, to pyruvate in the TCA cycle, and that's pretty self-explanatory. It's made in glycolysis and then it can be used in the TCA cycle. Next, we have the pyruvate coming down here and connecting the pyruvate and glucogenesis. This happens because the pyruvate from glycolysis gets carboxylated to oxaloacetate and then converted to PEP, and then eventually goes through the process of gluconeogenesis to regenerate the glucose. Okay, and then also down here with pyruvate, oh wait, no, we already did that one. We also have the glucose 6-phosphate, and that is connected up here in glycogenesis. And these two pathways, glycolysis and glycogenesis, share glucose 6-phosphate as a substrate. So in glycolysis, it's used for energy, and in glucogenesis, it is stored as glycogen. So, yes. And then, okay, next we're going to talk about the glycogenolysis, and we're going to start with the glucose 6-phosphate. This is a very important part of this pathways map because glucose 6-phosphate is a very common substrate amongst all of these, but we're going to start with how it is connected to the hexose monophosphate shunt. So we're going to follow the purple, and we go here. Basically, the same glucose 6-phosphate molecule produced in glycogenolysis can be diverted into the hexose monophosphate shunt if the cell is in need of NADPH or ribose, and the NADPH is indicated here with the yellow highlight. Okay, and then we're going to follow our second line back to glycolysis. So again, glycogenolysis and glycolysis share the substrate glucose 6-phosphate. And in glycolysis, it comes from the uptake of free glucose, and in glycogenesis, it comes from stored glycogen. And then lastly on this one, we have the glucose, and we're going to follow it up to glycogenesis. And if you notice, there's a double arrow on this one because this is a reversible reaction, an irreversible reaction. In glycogenolysis, the glucose, as you can see here, is an end product, and that end product is then used to help this process. So that is glycogenolysis. Next, we have the hexose monophosphate shunt. And so we're going to start here, glucose 6-phosphate. Once again, we're going to follow this over to glycogenesis. They share the substrate. Again, in glycogenesis, it gets converted to storage, and then in the shunt, it is actually used to oxidize. It gets oxidized, and it's used to generate NADPH, which we see here again highlighted with the yellow. And then secondly, we're going to follow this NADPH all the way to fatty acid synthesis, because it gets generated and can be used just in the process of fatty acid synthesis. It actually acts as an electron donor that turns acetyl units into fully saturated fatty acids. So I'm going to follow the light blue all the way down over here, and here we have the NADPH again indicated by the yellow, and my board is 2 separate boards, so that's a little messy, so I apologize. Okay, next we're going to talk about beta oxidation of a saturated fatty acid. And so here it can go 2 ways. In the first way, we have the excess acetyl-CoA that has been formed, and we're going to follow that red line down to the TCA cycle. And the CoA has a pink highlight because the micronutrient panthenic acid is a precursor of CoA, and that's actually vitamin B5. So that's why that is like that. And then down here we have this where it turns into a shortened saturated CoA activated fatty acid, and in that case it will be sent back up to the top of the cycle and re-enter the beta oxidation pathway. Okay, next we have the urea cycle, and here we're going to talk about the fumarate, and we're going to follow that pink arrow all the way down to the TCA cycle. And here the fumarate is a byproduct of nitrogen disposal. So once it gets transformed to maltate and then oxoalloacetate, it can then re-enter the TCA cycle and keep that process going and moving along. So next we're going to talk about the alanine glucose cycle, and we're going to start here with this glucose 6-phosphate and follow it up to the glucose 6-phosphate in glycolysis. Again, this is a shared substrate, and in glycolysis, the glucose 6-phosphate is the entry point for breaking down glucose to generate ATP, and in the alanine glucose cycle, the glucose 6-phosphate is the product of gluconeogenesis that you see here, and that happens in the liver, and then it can get dephosphorylated to free glucose and sent back to the muscle. Next we have this NH3 here, and as you can see, that is transported to the liver from the urea cycle. Oops, so we're going to follow that up to the urea cycle. Here we show the liver. So yeah. Next we have the pyruvate down here. So the glucose alanine cycle actually supplies the pyruvate that gluconeogenesis uses. So this happens after the pyruvate is turned to alanine and then turned back to pyruvate in the liver. So we're going to follow this pyruvate all the way. We're going to follow the orange line, and that is shown here in gluconeogenesis. Okay, so next we're going to talk about the fate of amino acid carbon skeleton. So here you notice that it can kind of end this way in the ketogenic process as ketones, or in the glucogenic process as pyruvate. So we're going to follow each one of those. So the ketones, we're going to follow the green arrow. We're going to go up to here to the acetyl-CoA in the TCA cycle. And the ketones can get transformed into acetyl-CoA and then enter the TCA, so that is why that connection is there. And then we're going to follow the pyruvate, the green arrow, all the way over here, again into gluconeogenesis, and that is the glucogenic pathway. And then we have this here. This is actually a TCA cycle intermediate, and this is alpha ketoglutarate. So we're gonna follow the green shown here, and this once it enters the TCA cycle, it is oxidized to CO2, as we see here, and then it is able to produce an AD and an ADH. Okay, so that is the end of my of me explaining all of how they're connected. But I do want to go back and talk about which pathways are related to which macronutrient group. So in the carbs chapter, we have glycolysis, glycogenolysis, glycogenesis, and glyconeogenesis, and then of course the TCA cycle as well. That one has to do with carbs, proteins, and fat. And then fatty acid synthesis, that was in the lipids chapter, as well as beta oxidation. And then we talked about the fate of an amino acid carbon skeleton, the alanine glucose cycle, and the urea cycle, all in the protein chapter. And so yeah, they're all to do with the different macronutrients, but they all come together to help our body metabolize. So yeah, thank you.